You may have heard of genetic engineering as a growing field, but have you ever heard of genetic super tomatoes? Genetically modified organisms and genetic engineering are crucial fields of science that is allowing us to make rapid developments in processes that have otherwise taken humanity thousands of years to develop. Just like how over this extended time, the produce we eat has been specially bred to maximize nutrition and crop yield, scientists are discovering new ways of using genetic technologies to accomplish this at breakneck speed. Welcome to the genetic basis of stuff and things. GMOs are organisms with DNA altered by genetic technologies, which is slightly different to our traditional crops. Our crops have been modified, but it's taken centuries of traditional cultivation methods, such as selectively breeding the crops with beneficial traits like yield and nutrition content. Now, barely any modern produce looks anything like their ancestors. GMOs, however, are able to produce beneficial results in rapid time. For example, genetically modified insect-protected cotton, maize, soybean and oilseed were shown to help reduce global pesticide use by around 22 million kilograms in the year 2000 alone. Think of how long that would have taken if they were picking and breeding individual plants. Another example is the tomato, one of the most economically important and widely cultivated crops globally. Our modern tomatoes, descended from the species Solanum lycopersicans cerciforme, are the products of selective breeding over centuries for specific sizes, shapes, and weights. For example, the San Marzano tomato was cultivated in Italy by selectively breeding tomatoes that were mutated into pear shapes. By the mid-20th century, however, our understanding of genetics was revolutionized by the discovery of DNA as a genetic material. This revolution fueled the development of technologies capable of rapid, artificial DNA transformation, the most famous being CRISPR. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats, and Cas9 is a nuclease called CRISPR-associated protein 9. CRISPR originated from bacterial immune systems, where it would recognize and eliminate specific viral DNA in the bacteria's genome. Thus, it is utilized to locate and digest specific target segments of an organism's DNA via double-stranded breaks, which is an unstable process in eukaryotes, meaning new DNA can be inserted into the broken genome with homology-directed repair. This utilization of CRISPR-Cas9 opened up worlds of possibilities, not only in removing detrimental genes from organisms, but also in inserting new beneficial ones. For example, one instance where modern CRISPR technology has been shown to engineer beneficial modifications is in inducing artificial parthenocarpy in tomatoes. Parthenocarpy is a development of a fruit without fertilization, resulting in sterile, seedless fruit. This is indeed advantageous for crop production. If a plant doesn't need to be fertilized, it doesn't need pollen, meaning it can go through harsh conditions where pollen is scarce. Seedless fruits are also higher in nutritional volume, since the seed cavities are replaced with pulp. Despite their seedless nature, subsequent generations can still be cultivated through breeding methods such as cutting, grafting, and cloning tissue. A study conducted by researchers in Japan demonstrated that it was indeed possible to utilize CRISPR to create parthenocarpic tomatoes. Here's how they did it. The researchers targeted a specific gene in the tomato's genome called CRLAA9. This gene is majorly responsible for regulating the plant's response to auxin, a key hormone that regulates signaling pathways promoting plant growth and development. Let me introduce two key players, ARFs and AUX-IAAs. First, ARFs, or auxin response factors, are transcription factors that bind to the promoters of auxin response genes and, when the auxin hormone is present, induce gene transcription. ARFs activate genes that promote processes that cause fruit development, including cell division. The second player, AUX-IAAs, or auxin-induced factors, is a repressor protein encoded by the CRLAA9 gene that interact with the ARF transcription factors, stopping the transcription of auxin response genes. We can see the relationship between all the protein factors here. Therefore, in order for the fruit development genes to be transcribed without pollination, either auxin must be present or the AUX-IAA repressors must be deactivated, which can be done by disrupting the CRLAA9 gene, because then it wouldn't produce any functional repressors. And this is exactly what the researchers did. They got to work constructing a CRISPR system with 3 different guide RNAs that directed the Cas9 nuclease to cut the CRLAA9 gene in 3 specified target locations. Once the first generation of tomatoes had grown, they tested their genotype using methods including restriction, fragment length, polymorphism analysis, and discovered that, depending on which guide RNA the CRISPR-Cas9 had used, up to 100% of the plants showed successful mutation into Parthenocarpic tomatoes. This is just one example of genetic engineering being harnessed to create incredible benefits. However, just like Spider-Man's Uncle Ben said, with great power comes great responsibility. This explosive development of genetic technology has the power to change all of life as we know it. Although luckily for us, GMOs are showing that we don't need to fear this area of science, since the benefits they reap have vastly improved upon various areas of crop farming and nutrition. So there you have it. Even the humble tomato can have a very important role to play in advancing the frontiers of science.